This right here is a test. It's something that all of us have to take many, many times during elementary school, high school, and post secondary. Like viruses, tests come in all shapes and forms, some on paper, some online, some in the form of practical experiments. But what if I told you there are some tests that can save your life and the lives of others? No preparation needed, no cramming, no note taking, no scantrons, and no essays. And in today's episode, we will talk about tests and screening methods for the coronavirus and HIV, which are the first important steps in helping those infected by these viruses. By now, you're probably familiar with my face. I'm Mathab, and this is episode 4 of 5, sponsored by the Center for Blood Research at UBC. As we saw in our first episode, since 1981, worldwide, nearly 72 million people have contracted HIV. About half of those have died, and there are now about 39 million people living with HIV. In 2014, the United Nations Program on HIV/AIDS unveiled their ambitious goal of ending the AIDS epidemic by 2030. For this, they set a target for 2020 called 90-90-90. This means that 90% of the people living with HIV know about it, 90% of those diagnosed will receive treatment, and 90% of people following treatment will have viral suppression, which means the treatment manages to lower the HIV to a point where it's undetectable. These targets have not quite yet been reached everywhere, but overall we've made good progress. The first and most important step is diagnosing HIV. Without this, people don't know they need to get the treatment. Another important reason for diagnosis is that if you know you have HIV, you can take steps to protect yourself and others and make it less likely to pass it on to someone else. Okay, usually when you're sick, you can go to the doctor and they can sometimes even diagnose you on the spot. But diagnosing HIV isn't as simple because the symptoms are quite general. What I mean by that is that earlier in an infection, for a few days, you might feel like you have a flu, so have something like a headache. You might experience some soreness in your arms or legs, or you may have a fever. Later on, you might not experience any of these symptoms at all. So, how do we know whether someone has the virus? Well, we need to run some tests. There are 3 main things we can look for to see whether someone is infected with HIV. We can look for the presence of antibodies against HIV. We can look for viral proteins or parts of the virus's genome in the body. Before moving on, I just want to mention that if you think you've been exposed to HIV, you can be tested through the anonymous HIV testing program offered by the BC Centre for Disease Control. It's very easy to get tested, and through this program, you don't have to give up any identifiable information about yourself. It's totally anonymous. There will be a link to this program in the description for this video, which lays out the steps to get tested. Let's talk about antibody testing first, which is the most common way of diagnosing the virus. If you recall from the last episode, antibodies are created by specialized immune cells called B cells to destroy pathogens that enter our body. From the time HIV enters the body, it takes a few weeks for the B cells to make antibodies specifically against the virus. The HIV antibodies can be found in many different fluids within the body, like the blood, which is often used for testing because it's pretty easy to get. Now, if antibodies against HIV are detected in the blood, it means that you have been infected with HIV. How do you check whether someone has the antibodies for HIV? Antibodies are looked for in our blood through a special technique called ELISA. Really small quantities of blood are first processed in a way which leaves us with only things like proteins, DNA, antibodies, and the virus, if it's there. This sample is then placed in something called a plate, and this plate has many little wells that look kind of like this from a side view. Okay, each well is lined with HIV antigens, which are the protein traces of the virus. It also has detection antibodies, which are meant to bind to HIV antibodies in the blood, if there are any. It's interesting how we're using something that exists naturally. In research, we end up using many natural proteins to help us in the lab, and antibodies are just one example of this. So let's say we get a sample of blood and put it into this well to see if the person has antibodies for HIV. If you remember, antibodies are supposed to bind to specific antigens, which is why the detection antibodies don't bind to the HIV antigen and nothing happens. So if nothing binds to the antigens, that means there are no HIV antibodies. Therefore, this is a negative test result. It is highly likely you do not have an infection. There's a chance you might get a negative test if it's early on in the infection. So if you think you're at risk of being exposed, it's advised to take a test again after 3 months or so to give time for the antibodies to develop. In another scenario, we get a sample of blood and place it in the well. Now we can see the antibodies against HIV bind to the antigens. We can also see the detection antibodies connect with these HIV antibodies. When this happens, the color of the well changes. And this color change indicates to us that there are HIV antibodies present in the blood. This color change means that it's likely there is an HIV infection. And so that's called a positive test result. Okay, at this point, if a positive result is received from the antibody test, we need to double check to make sure that it is HIV that's causing this. This is because other conditions, like something called lupus, you may have heard of that before, or a bacteria called syphilis, can cause this test to produce a positive result. So to double check if there is an HIV infection, we need to run a protein test. Okay, so we do this protein test to confirm the positive result from the ELISA. The test uses a technique which helps sort the proteins by their size. It does this with a kind of gel-like jello that looks a bit like a castle wall. What we are looking for here are specific HIV proteins. And here's HIV, which I'm sure you're getting more familiar with by now. 2 episodes ago, we talked about the structure of HIV and the different proteins in them, like the glycoproteins, integrase, there's reverse transcriptase, and protease. Hopefully, some of these ring a bell to you. We can take these proteins and put them on top of a gel with a dye and a bit of electricity to help them along, and they begin to move through the gel. It's a bit like throwing objects at a bunch of nets. Smaller things like marbles would go through and go further than, say, a tennis ball. It's the same idea here, where the smaller proteins move further along the gel. We take samples from the ELISA and do the same thing. If the pattern matches up with what we know are HIV proteins, then it means the sample is positive for HIV. Okay? If the pattern is different though, it shows the sample had different proteins, not HIV proteins, and so is HIV negative. Let's move on to the third category of testing. If you remember, HIV has an RNA genome. So if we detect HIV's RNA in the blood, that means there is an HIV infection. HIV's RNA is detected through something called a polymerase chain reaction, or PCR for short. This test can detect very low levels of HIV RNA in the blood and then amplifies it to make the presence of this RNA very clear to us. By amplify, I mean making many copies of this piece of RNA. We can compare this to a game of Where's Waldo, where instead of finding Waldo, he gets multiplied into millions and millions of copies, and that makes it much easier to find him. For this amplification to occur, however, the RNA of the virus must be transformed into DNA through an action called reverse transcription. We actually use proteins, much like HIV, to convert RNA to DNA. We also need short snippets of DNA called primers that match the specific DNA that we're looking for, so we can amplify it. If these primers attach to the viral DNA, they start a chemical reaction that allows the DNA to be copied or amplified. And we can see the reaction in motion here. We can see the DNA being zipped open, and once the primers attach, more and more copies are being made. This reaction is repeated over and over, each time multiplying the number of DNA segments. This is all possible thanks to a machine called a thermocycler, which is basically just a fancy oven. The thermocycler controls the temperature and allows for the DNA to be separated, the primers to fit to the DNA, and for the DNA to be amplified. This machine, and PCR in general, is not only used for detecting HIV in someone's blood, it has many other uses. We actually use it in our lab. And in our lab, we study the health of mitochondria following HIV infection or after taking treatments for it. You may know the mitochondria is the powerhouse of the cell, but did you know the mitochondria have their own DNA? They do, and that's pretty cool. We also look at the ends of chromosomes called telomeres. Both of these things tell us about the health of a cell, but in order to get that information, we need to amplify these two types of DNA using PCR. We have to mix the sample we are studying with necessary elements like primers. This mixture is then spread into a plate and inserted into the thermocycler. Based on how much of the mitochondrial DNA and telomere DNA are amplified, this gives us information on the health of the cells, which is important for our research on aging. Let's move on to how diagnosing SARS-CoV-2 works. The main way of testing for the coronavirus currently is by doing a nasopharyngeal swab. This long swab is inserted through the nose and scrapes the back of the throat to try to pick up any viral particles that may be present. Like HIV, the coronavirus has an RNA genome, so we use proteins to once again convert the RNA to DNA through reverse transcription. A PCR reaction is then performed to detect any virus RNA that may have been picked up by the swab. It's so sensitive that even just one particle is enough to know that someone is infected. If the swab picks up the virus, the DNA will be amplified, showing a positive test result. If not, there will be no amplification at all, which means the test was negative. As we've seen previously, our immune system responds to a viral infection by producing antibodies that are specific for the virus. Like for HIV, SARS-CoV-2 antibody tests have been developed to detect specific antibodies in the blood against the coronavirus. We've now seen different methods of testing, but how do we know which one to use? This graph shows how likely you are to detect a viral infection depending on when we're likely to detect the virus's RNA, which is that green line, and antibodies against it, which is in blue. So starting from the point you get infected, it takes about 2 weeks for most people to start experiencing symptoms, if they experience any at all. As more and more viral copies are made, the amount of viral RNA in our body increases. After a couple of weeks, the amount of viral RNA decreases, and that's because the adaptive immune response is kicking in, and specific antibodies are being made by B cells to combat the virus. The levels of viral RNA, protein, and antibodies change with time, affecting the results of tests, depending on what type of test you use and when you use it. A simple way to think about this is that RNA tests are good early and tell you if you have a virus right now. Antibody tests are not good for early diagnosis, but will tell you if you've ever had the virus, and can also help you if you have some immunity to the virus. This is different for all viruses, however. For HIV, having antibodies doesn't mean you're immune. For SARS-CoV-2, we don't know yet, but with more testing and more research, we may get that answer pretty soon. Overall, tests are very useful and very important. It could save your life or that of others around you. When it comes to HIV, testing needs to be a priority in order to know who has the virus and be able to start treatment as early as possible. This will help us reach and exceed the 90-90-90 goal and help save lives. The earlier the HIV treatment starts, the better it works. Also, early diagnosis helps prevent transmission. When people know they're infected, they can take steps to prevent the spread to others. And that's the case for the coronavirus as well. RNA testing for COVID 19 is and has been important for identifying those with the coronavirus early on and tracing them so we can prevent the spread of the virus. This is a big reason why BC has been doing relatively well. We may still be a ways away from returning to normal, but testing might help. With antibody tests, knowing how much of the population has already been infected, how many people may then have developed immunity, and for how long, will be useful in moving forward as a society and understanding the impact of the COVID 19 pandemic. It may also give valuable information for efficient prevention and treatment measures, which is the topic for next time on our last episode. Until then, that is it for episode 4. As always, I want to leave you with a question. Which of the following would not likely help in treating HIV or the coronavirus? So, 3 of the pills does not likely help in treating either of these viruses. We'll answer that next week. Thanks for tuning in.